First, thank you so much for this great opportunity that we can present our ongoing study to show the novel microbiome signature in the HIV elite control. So a little bit, here. A little bit of brief background. Elite control represents a unique group of HIV who has ability to maintain undetectable plasma HIV viral load without ART. And therefore, this elite control, referred to EC, has been used as a model to identify the novel therapeutic strategy and also for the functional HIV cure. And among other factors, the gut microbiome has been reported to different between the elite control and the HIV negative treatment negative patient and also the healthy control. And among the few studies which has used the 16S RNA gene sequence, which we have reported in the recent literature, and has shown that elite control, they have a richer bacterial microbiome profile compared with treatment naive patient and also the healthy controls. And among different studies has reported different abundant bacteria that are differential abundant between the different groups. So the aim of our study was to use the deep shotgun metagenome sequence and use the up-to-date pipeline to characterize the microbiome at both the species but also strain level. In contrast, the previous study used the 60S, which are limited in bacteria. We know that the genus level at most cases. And including and also the uncharacterized microbiome, which has been in a recently a very interesting study show that when using this new pipeline that include uncharacterized microbiome, the strongest biomarker that's associated with the diet in the mice is actually uncharacterized microbes. So we assume that this simultaneously might also happen in the human gut in the context of human health and disease, and also in this EC context. And also we want to study the microbiome genes and also the proteins and pathways against different databases in correlation to the EC phenotype. And in this study, we have performed a deep shotgun metagenome sequence over 50 million reads per sample from a well-defined elite control cohort and also including control both the HIV treatment naive HIV patient and also the HIV negative healthy control. And we collect the key demographic and clinical data, including the age, gender, origin, and also transmission model or sexual behavior, which has been reported to linked with HIV-specific gut microbiome dysbiosis. So, and then we use the updated pipeline to study the microbiomes at the genus level and also study the genes and the pathways using different databases and also the bioinformatics and the correlation to identify the EC-specific biomarkers and both the taxonomy and also functional level. So our main study first, we observed as expected that elite control, they show a significant enriched microbiome alpha diversity, which means that they have much high unique bacteria richness in the elite control as compared with the patients. And also, as mentioned, that the transmission model has been linked with HIV-specific gut microbiome dysbiosis. So in our study, we try to stratify our study cohort based on the transmission model. And in our study, our participants are predominated by the heterosexual individual. So when we look at this group, we see the same tendency actually stronger significates the alpha diversity in the elite control as compared with healthy with the patient. And also, the healthy control, they have higher diversity as the patient. And then we look at the beta diversity, we see the overall microbiome composition, considering both the unique species, but also their composition and balance. And we see that elite control, they have significant different composition compared with the patient and also the negative healthy controls. And then the next, we want to understand what microbes has been different between the groups, either enriched or decreased. And very surprisingly, if you look at the left corner, we list the top significant species. And those marked with GGB, they are actually just the genome assembling without any no species. And so our study showed that when we look at the whole picture, whole landscape, the strongest elite control-associated microbiome species, they are actually either uncharacterized or very newly reported species which function are rarely studied or not studied at all. And then, as I said, that several factors can impact microbes. So we have used the regression models try to adjust the different covariants, even though this study sample size is quite small. And after adjustment of all the covariants, we find that the 5 tags, they are significantly enriched in elite control as compared with the patients. And among those, 2 are unknown species. Yeah, even actually unknown the genus or the family. And the other three they are just recently reported, which there is no study about the function. And actually, the first one, there is a study that showed these bacteria, they can protect the metabolic disease in the mice models. And otherwise, there is no functional study. And also compare with the elite control and the HIV negative control. And we also observed 4 significantly enriched tags in the elite control. And I also want to highlight that the top 3 that I highlight here, they are enriched in EC as compared both to patient and also the health control. So the main take-home message is that the gut microbiome in elite control, they are significantly different from the patient and also the HIV negative control. And the strongest EC-specific species, they are novel species, either uncharacterized or recently identified, whose functional are rarely studied or not studied at all. And now we are doing the work in progress is we are performing the function analysis, as I said, using different database protein pathways. And also very interesting, we observed that the top significant proteins enriched in EC, actually they belong to the immune inhibitor, also the peptides, which I haven't shown here now because we are doing the rigorous data filter to make sure that these proteins are from microbes and not the humans. And then the next question, I think, is always endpoint question that to answer how the microbes that they could possibly contribute to this HIV virology control. We have 2 main hypotheses. First, the microbes may produce some metabolite that can regulate the human immunity, and it's well known that there are numbers of the microbiome-derived metabolites. And secondly, these specific species, they might produce the metabolites or the peptides with potential anti-HIV effect. And actually, we have indeed identified anti-HIV peptides, which my colleagues, Rafa, next speaker, will present us in detail. And even though we have no clear evidence say that these D peptides are from the gut microbiome, but they are indeed enriched in the fecal sample from elite control. And we know that bacteria, they are well known to produce the D peptides. So we are really excited to dig into further steps. So I'd like to thank our project team and also thanks for all your attention. Thank you.